students, welcome to Biosastra Talks. I hope all of you are doing well, and we are going on with the next video in this session, like the paper 45 Q&As that we are going on in the previous sessions. And in this video we are discussing some other questions like from 136 to 150. So let's get into the topic. Okay. So what was the next question that given from the NEET paper code 45 from biology session is it came from the actually DNA like genetics chapter and from the like recombinant DNA and everything. If you see the question, what is the question is statement 1. Here in the statement 1 they are saying that the DNA fragments extracted from gel electrophoresis can be used in construction of recombinant DNA. So here they are saying that DNA fragments that were produced from gel electrophoresis are used in the construction of the recombinant DNA. And in the statement 2 they are saying that smaller size DNA fragments are observed near the anode and larger side are found near the cathode. Okay, in these two you have to say that which one is like correct and which one is wrong, or they both of them are correct, or like that. Okay, if we see that they have given some of the options for us, like in that you have to say both of them are correct or both of them are incorrect and either one of them are correct. Okay, let's see what is that. So if we see the gel electrophoresis or recombinant DNA, normally what happens is we know that we'll take a gel, agarose gel, and then we'll drop the DNA samples from here. So what will happen when we drop the DNA samples here? The DNA molecules, normally we know that there are so these strands are nothing but the DNA molecules. In that DNA also there are some small strands and there are some larger strands. Okay, so normally we know that the smaller ones can move fast, right? So like that only, larger, like if we see according to the humans, the like uh, the children can run fast when compared to the adults, right? Like that only, the smaller DNA fragments can move fast, so that what they'll do, they'll move fast towards the anode here, which is like positive thing. Okay, so they move up to the towards their what they'll say charge, and they move towards the anode. Why? Because they are smaller in size, whereas the larger fragments cannot move that much fast. So that what they'll do, they'll place at the walls of the agarose gel. Means somewhat you can see that these larger fragments are present near the wall of the agarose gel. So that's what here happens. So according to this second statement, the smaller size DNA fragments are observed near anode only. Okay, why? Because they are negatively charged. Okay, whereas the larger fragments, they are near the wells or walls of agarose gel, or you can also say wells of agarose gel. Here you can see the larger fragments. Okay, so this statement is correct. If we move on to the first statement, what they are saying that these fragments extracted were used in the recombinant DNA. Yes, why? Because we know that normally why we produce these why we'll do the agarose gel electrophoresis means to separate the smaller and larger fragments and to separate the DNAs that we need. Actually we'll have a solution of DNA A and B here. So in that we need the only DNA A molecule. So we know that this DNA A combination, what we'll do, we'll arrange the gel according to that combination. Obviously then these DNA A molecules are separated from the B, so we can extract these DNA molecules in separately and then go on with the recombinant DNA technology with our specific gene. Okay, the DNA which is containing the specific gene. So the first statement is also right, that the DNA fragments extracted from gel electrophoresis are used for the recombinant DNA technology itself for the production of more amount of DNA. Okay, so here both the statements are correct. Okay, and it is also correct explanation. Statement 2 is also correct explanation for the statement 1. Okay. So here you can say that the option 1 is correct. Okay. And we'll move on to the next question. What is next question is they have given the two lists here. This is according to the molecules that are present. So in that, first one is adenosine, adenylate acid, adenine and alanine. We know that what are they. So we'll move on to this. Normally if you see that what is an adenosine, adenosine is like, if you see the picture here, you can easily identify adenosine is nothing but a nucleoside which contains, nucleoside means what, which contains sugar and nitrogen base, right? If we see the DNA structure and everything, this adenosine is normally like we'll have nucleotides and nucleosides. First thing, we'll have nitrogen bases and then and then sugars and then phosphate groups, right? So in these, nitrogen bases are separate, and nitrogen base and sugar is separate, and three of them are separate. So, whereas nitrogen base and sugar are known as nucleosides, okay, without phosphate group are known as nucleosides, whereas three of them combine to form nucleotides, okay, whereas only this one is known as like no, only nitrogen base is known as nitrogen base, that's it. Okay, so like this you can separate, or you can differentiate the nucleoside, nucleotide, and here. So here in this option they have given so many things. So what is adenosine actually? If we see here, adenosine is normally, by the picture you can say that it is a nucleoside. Okay, why? Because it contains the adenine and sugar. Okay, it may be like ribose sugar or deoxyribose. Here is actually it is a ribose sugar itself. Okay, why? Because we are containing the oxygen and everything. Okay, and another thing is the next one, adenyllic acid. Adenyllic acid means I already told you that which contains the nitrogen base, sugar, and phosphate group. Okay, this is known as a nucleotide, the full combination which is present in the DNA and RNA molecules, right? So this is the nucleotide obviously, which contains, a like adenosine nucleoside and adenyllic acid nucleotide. Okay, so if we move on to the next one, adenine, they are asking. Adenine means nothing but we know that we are having 4 nitrogen bases: adenine, guanine, cytosine, thymine. So adenine is obviously the nitrogen base which is present in the nucleic acid. So here adenine is the nitrogen base which contains the nitrogens. Okay, whereas the alanine, it is totally different from all these three. Why? Because if you see that normally alanine is an amino acid which contains both amino group and acid group. So the amino acid which contains the methyl group as a side chain in the place of R, it is known as alanine. Okay, so like that, if we see the combinations, these are the combinations. So what was the correct option here is A3. Okay, three options are there for A3, adenylic acid B2, and these two are having B2, whereas C1, okay, this one is having C1 and D4. Okay, so the third option is correct here. Okay, we'll move on to the next question. What is the next question? It came from the female and male gametogenesis and everything. If we see here, they are asking about the reductive division of the human, and they have given some statements, and you have to say which one is correct and which one is wrong. The first statement is reduction division of human female gametogenesis starts earlier than that of the male gametogenesis. Okay. Second one, the gap between the first meiotic division and the second meiotic division is much shorter for males compared to females. Okay. Third one, the first polar body is associated with formation of primary oocyte. Fourth one, luteinizing hormone surge leads to disintegration of endometrium and onset of menstrual cycle. Actually, it's not right. It is not right. Okay. Normally here the follicle stimulating hormone will play a role. So this is wrong answer. We'll move on to the remaining options here. You have to say which two are correct. Okay. So D is obviously wrong. So we can consider these three options like A, B and 3, A, B, A and C and B and C. So the first polar body, like if we see the answer here, normally if we see the image here, we know that what happens in the male and female is in the male obviously the parental spermatogonia undergo which are having the diploid condition, they'll undergo meiosis 1 and then meiosis 2. Okay, when in within fastly, okay, we'll see that they'll undergo meiosis 1 and meiosis 2, and then after the meiosis 2 some of them will produce the sperms which are having the 2n condition, and some of them will have the sperms which are having the n condition. Okay, here these sperms are haploid, and these are you can see that some of them will have both parental characters like that in the meiosis 2. Okay, they'll have two mature sperms which are having the spermatogonia and everything. If we see the female one, this is known as oogenesis. Here also the meiosis will take place. In the meiosis 1 itself, you can see the formation of the first polar bodies. Okay, in the first one will have equal thing, and the second one for only one daughter, or one offspring will show all the chromosomes and the other will not show it. So in the second polar body you can see that the mature oocyte is formed from any one of them and from in both the characteristics. So see here you can say that the option both of them, the option A and B are correct. Why? Because normally it will takes more time when compared to the female one. Okay, so the primary oocyte is not the primary polar body does not produce the oocyte, no. So this is the option 3 is wrong there. Okay, option C, if you see the question, so the option 3 is wrong, whereas option A and B are correct from this question. Okay, we'll move on to the next question. All living members of the class Cyclostomata are, here they are asking about the class Cyclostomata. Normally we know that maximum Cyclostomata means the organisms which are nothing but the ectoparasites. Ectoparasites means which will depend on the host directly from the outside. Okay, ecto means outside, not endo. Here you can see that this is the ectoparasite. Okay, so what it will do, this is doing the gillfish, what it will do, it will depend on the fish from the outside itself, means it will depend on the normal fish for its purpose. Okay, so normally the all members of the cyclostomata are ectoparasites on some fishes. Okay, you can say ectoparasite means what it will do, it will attach from the outside and will affect the fish. Okay, so a parasite means what is that, the organism which depends on the other host, okay, for its living, and it will affect the host, like that only this ectoparasite will affect the fishes in some ways. Okay, so here what is the answer is the all the members of cyclostomata are obviously ectoparasites. Okay, we'll move on to the next question. What is the next question is, they have, this one is also from the statements itself, it belongs to the Golgi apparatus and its function. So if we see that what is the primary, first in the first statement they have given that the primary function of the Golgi apparatus is a package of, one minute, is to package the materials made by endoplasmic reticulum. Okay, endoplasmic reticulum, and deliver it to intracellular targets and outside the cell. Okay, first statement. Second statement, vesicles containing materials made by endoplasmic reticulum fuse with cis phase of Golgi apparatus and they are modified and released from trans phase of the Golgi apparatus. Okay, these two statements they have given like assertion and reason. In this you have to say both of them are right or both of them are wrong, or right and correct explanation means reason is right and it is correct explanation for the assertion or not. Okay, if we'll see the picture you can understand it easily, the answer. If you see here you can observe that these are the Golgi bodies that were formed and this is the endoplasmic reticulum that is present on the cell wall of the nucleus. Okay, this is nucleus, right? So we know that there are two types of endoplasmic reticulum. These are the rough endoplasmic reticulum. Okay, so these rough endoplasmic reticulum, if we see that from the question here, if we see that what they are saying that in the first question here what's happening was normally the vesicles, okay, normally the materials which were produced from the endoplasmic reticulum were transformed, like they are transformed from the endoplasmic reticulum through the transport vehicle into the some place of Golgi apparatus, which is nothing but the cis phase. This phase is this side is known as cis phase. So these vesicles are transported into the cis phase of the Golgi apparatus, and then they are transferred from cis phase to edge of the Golgi apparatus. That is nothing but, here you can say that this is the trans phase. Okay, so means in the second option, the materials which are produced from the endoplasmic reticulum will enter the cis phase and they'll travel through the Golgi apparatus and they exit either into the intracellular spaces, they are present within the cell, or they'll exit outside the cell. Okay, so the second option is correct here, whereas from the first option also you can see that normally the food materials, here in the assertion is also true. Why? Because the Golgi apparatus functions to package, right? Why? Because the Golgi, what it is doing here, it is packaging the food materials that were produced from the endoplasmic reticulum. So and it is and transferring transferring them to various destinations. So assertion is true and reason is true, and obviously it relates to both. In this reason also they are saying about how it is transferring the material. So here the assertion and reason are true, and the assertion and reason is also correct explanation of the assertion. Okay. That's it. We'll move on to the next question. What is the next question? Match the list 1 and list 2. Here they have given some of the questions from the seeds itself. If we see the images here, you can understand easily. Here they are asking, they have given the layers, and you have to say whether they are present in the seeds or not. First one is scutellum. Normally you can see the scutellum in the monocot seeds itself. Okay, we know that the scutellum is present in the monocotyledons only, not in the dicotyledons. If you see the this is the dicotyledon, that is nothing but the groundnut seed. Here you cannot see the formation of scutellum. Okay, scutellum is a layer which is present only in the monocotyledons. Okay, first one is this. And non-albuminous. Non-albuminous means normally the monocotyledons are albuminous in nature and which contain the endosperm. Okay, so like sorry. Yeah, right. So non-albuminous means which will not contain the endosperm. So here you can see that. One minute. Yeah. So they are nothing but the non-albuminous are nothing but the seeds actually. They does not contain endosperm actually. Okay, no endosperm. So these are the albuminous. Sorry, I have confused. So these are albuminous seeds which contain the endosperm layer which is responsible for the seed growth, whereas the non-albuminous seeds does not contain endosperm. Here you can see that this is the non-albuminous seed, the groundnut seed. It does not contain any endosperm. Okay, so here non-albuminous is nothing but the groundnut which does not contain any endosperm. Okay, if we move on to the epiblast is nothing but you can say that it is a rudimentary cotyledon. Normally the edge of the cotyledon, here you can see that normally in the monocotyledons they are present. They are known as epiblast. The edges of the cotyledon are nothing but epiblast. Okay, perisperm is nothing but a persistent nucellus. Okay, which is present only like here you can see. Okay, like perisperm is present in the persistent nucellus of some seeds like black pepper, beetroot, and etc. Okay, so these are the correct options. So what is the correct answer here? A 2. Okay, B like non-albuminous 3, B 3, A 2, and C 4. D1. Okay, so option 1 is correct from this question 141. We'll move on to the next question. Given below are two statements. Again, it came from the statements itself. Like it belongs to both vertebrates and chordates. So here what they are asking is all vertebrates and chordates. All vertebrates are chordates, but all chordates are not vertebrate. Okay, and whereas the reason the member of subphylum vertebrae possess not notochord during the embryonic period, the notochord is replaced by a cartilaginous or bony vertebral column in adults. Okay, so in this you have to say which one is correct and which one is wrong. If both of them correct, and they are correct explanation. If you see here, normally this is a chordate which is like which will develop into a vertebrae. Okay, normally if you see here why they are known as chordates means they'll contain the notochord layer. Okay, this notochord is present in the chordate members, and then this notochord is developed into vertebral column in the evolution process. Okay, so normally so you can say that all vertebrates are chordates, but all chordates are not vertebrae. Okay, so here according to this, and the second reason you can see that the member of subphylum vertebrae possess the notochord, which first of all the vertebrates first of all they contain the notochord itself, and then it is developed into a specific vertebral column which we are having here, like backbone. You can say that like it lines itself like in the backbone of humans, animals, and everything. So this notochord only, first of all the chordates will have the notochord. In the specific evolution it is formed into vertebral column or the backbone. Okay, so like that obviously reason is correct here, and also the answer like assertion is also correct here, and you can say that both assertion and reason are correct. That's why it is the correct explanation. Why? Because normally it is replaced by the notochord, no, so obviously they are not chordates itself. That's why this is the correct explanation. Why? Because it is replaced by the cartilaginous or bony layer. Okay, so that's why it depends on two layers. Here A and R are true, and it is the correct explanation of A. Okay, and another thing, why? Because they are saying that why all the chordates are not vertebrae. Why? Because some chordates will not develop into animals, right? That's why. If we move on to the next question, it came from the antibody structure itself. Here they are asking the statement that is not correct. If you see the statements here, the first option is each antibody has two heavy and two light chains. Obviously here you can see that light chain and heavy chains, right? These are the light chains. Okay, you have to say whether they are two or not. Okay, and sorry, I have get the answer itself. Okay, so what will happen is if you see the structure here, you can say that these are four light chains. Actually you can see that here two are there, and this side two are there. So total four light chains are there. So this option 1 is wrong. Why? Because four light chains and two heavy chains. So the option 1 is wrong. The heavy and light chains are held together by disulfide bonds. Normally the light chains are held together by disulfide bonds, that and heavy and light chains are also held together by disulfide bonds itself. Okay, this is right. Whereas third statement, antigen binding site is located at the C-terminal region of antibodies. If you see the C-terminal region here, normally the antigen binding site is present normally here in the N-terminal region, not in the C-terminal region. Okay, here the antigen will combine at here, actually N-terminal region, not in the C-terminal region. So this is also wrong. Like antigen binding site is located at the C-terminal region of the antibodies, but normally it is present at the N-terminal region. Okay, if we see that constant region of heavy and light chains are located at C-terminal, so obviously the constant region is present in the C-terminus of the antibody. Why? Because they both combine at the C-terminus itself. This is right. So in this question, normally I forgot to give the options. Actually these two are wrong. Like the 1 and 2 are the wrong, no, correct answer, like wrong answers. Like you have to say 1 and 2 are wrong from these statements. Okay, why? Because it contains four peptides, four light chains and two heavy chains, and the heavy and light chains are held together. Sorry, 1 and 2 kaadu, 1 and 2, not 1 and 2, 1 and 3 are wrong here from this question. Okay, so another thing, silencing, it came from the silencing of specific mRNA. You have to say it. Normally we know that the mRNA which is produced from the transcription process, it is silenced somehow. Why? Because silencing means, normally we know, simply if for example, normally we are talking in a group, when the other persons don't need us to talk, like what they'll do, like for example, simply we'll say that the elders will be there. What they'll do when we are talking nonsense or we are talking any bad words, what they'll do, they'll tell us to stop or they'll tell us to silence. So like that only, normally the silencing of the genetic materials will also takes place in our body. How? Means normally the silencing of the mRNA will takes place here. The mRNA molecules takes place via RNA. Why? Because they are asking because of these double-stranded RNA. The double-stranded RNA, we know that double-stranded RNA are not what they'll say, they are not stable. Okay, what they'll do means these double-stranded RNA, obviously they are present in the eukaryotic organisms as a method of cellular defense. Normally this double stranded RNA or RNAi is the RNA which is present in all eukaryotic organisms as a method of cellular defense. Okay, so normally what it will happen is, this is for the cellular defense itself. What it will do means, sometimes this RNAi, it will combine with some of the disers and other breaking compounds, and is attached to the mRNA produced from the transcription, and it will silence that gene. Silencing means it will break down that mRNA into pieces so that it cannot code, right? If the mRNA is full, then it can code into another protein, but in it is broke down into so many pieces, it cannot code for any any specific protein. So like that it will silence the mRNA by with the help of the RNAi or double stranded RNA, or you can also say it is known as a complementary double stranded RNA. Okay, so the complementary double stranded RNA will help in the silencing, not help, it will induce the silencing of the mRNA, which is normally this complementary double stranded RNA is present for the self defense of our organisms, for the any from the parasites or somewhat. Okay, we'll move on to the next question. What is next question is genes R and Y. It came from the genetics part itself again. Actually, in this paper they have given so many questions from the genetics and ecosystem and plant kingdom also. Okay, so if you see here they have given the question that genes R and Y follow independent law of independent assortment. If RR produce round, RRYY produce round and RRYY produce wrinkled green seeds, what will be the phenotypic ratio of F1 generation? Okay, here they are asking that it is nothing but law of independent assortment is nothing but the dihybrid cross. Okay, so you have to say that what will be the phenotypic ratio. If you see here, this is RRYY, we know that normally what will happen by this, from the first generation itself we'll get the combination of capital R small r and capital Y small y, that is nothing but round wrinkled. Okay, heterogeneous round wrinkled. So when these RRYY will go with the F2 generation, F2 cross, we can see that so many combinations of the seeds were formed here. Some of them will be round and yellow like this one, round yellow, round yellow, round yellow, round yellow. This one is also round yellow, and this one is also round yellow, and another thing, this one is also round yellow, this one is also round yellow, this one is round yellow. 1 2 3 4 5 6 7 8 9. So these are all round yellow seeds, and again round green seeds. Actually, if you see, round green means actually this one is round green, this one is round green, and this one is round green. So total 3 round green or seeds. And another thing, wrinkled yellow. Wrinkled yellow means here you can see that this one is wrinkled yellow, this one is wrinkled yellow, and this one is wrinkled yellow. Another 3 will be there. And last but not least, what is that? The wrinkled green will be present, only one is evolved. So when we cross this like RRYY, like capital RRYY, and like homogeneous seeds, with when they undergo the F2 generation, they'll produce heterogeneous combinations of seeds, like with the perfect combination of 9:3:3:1. Okay, so that's it. 9 means 9 round yellow, 3 round green, and 3 wrinkled yellow, and 1 wrinkled green. Okay, we'll move on to the next one. What is the next question is, it came from the proteins, like from the genetics itself again. So it is like histones are enriched with which type of amino acids they are asking. Okay, here answer also came before. So according to this, you can say that lysine and arginine. If you see the picture, you can analyze that there are like they have given these dots are nothing but the lysine molecules which are present. This is the histone protein that is present in the DNA. Normally we know that DNA are coiled with the histone proteins H1, H2, H3, H4, right? They are like for the coiling itself. So this is coiling, and these are like the strands that were coming, and this is the DNA actually that is coiled again. Okay, so here if you see that these histone proteins contains, these pink ones are nothing but the lysines, and whereas the green or empty ones are nothing but the arginine. So normally the histone proteins are rich in lysine and arginine amino acids. Okay, we'll move on to the next question. The first menstruation. Normally we know that menstruation takes place in the females. So here they are asking what is the first menstruation is known as. Normally menstruation occurs in 3 stages, like first menstruation is known as menarche, whereas the menstruation that occurs in the females which were like about 12 to 50 years are known as menstruation, and after 50 years you can see the menopause. Menopause means stopping of menstrual cycle. Okay, so that it will lead to the like death of, like after 50 years when the menopause is stopped, according to that the female's body is changed. Okay, so it will happen. So the first menstruation is called as obviously menarche. Here you can see another option is also diapause. Diapause is nothing but it is a condition that is seen in insects. We know that what it will do, in this condition the metabolic activities of insects are stopped. Okay, that is known as diapause. Okay, it is seen in the insects, not in the humans. Whereas ovulation means we know that the formation of eggs, okay, during the menstrual cycle. So this is not called as the first menstruation. So answer is menarche is known as the first menstruation that normally seen in 11 to 12-year girls. Okay, when it starts, normally it starts at this age. It may be after 12 years also it will occur, but for starting age is like 11 to 12 or something. Okay. Next question. What is the next question? Here is 148. That is here they have also given some matching lists. You have to say like heart and everything. And if you see that options are there like heart, kidney, gastrointestinal and adrenal cortex. And here they have given some of the complexes like compounds like erythropoietin, aldosterone, atrial natriuretic tract factor and secretin. Okay, you have to match them according to the organelles they are present. If you see here, normally first of all we start with the heart itself. Normally the heart contains the atrial natriuretic factor. What it will do, what is the role of this factor is actually it is present here. You can see this green line, right? So this is the atrial natriuretic factor. What it will do means it will maintain the sodium and water balance in the heart. Okay, so it will maintain the sodium and water molecules that were flowing through the heart. It will maintain the sodium and water balance. Why? Because normally what the doctors will say, like when we'll go on for the BP checkup, what they'll say, if you are having the high BP, what they'll say, eat less salt. Right, we know that the salt contains sodium chloride. So like that only, according to that we know that the sodium content is high means blood pressure will be high. So for maintaining that sodium and water content in the heart itself, this atrial natriuretic factor will function. Okay, it appears like this. What it will do means it will allow the sodium or water from the kidneys, liver, etc., or the lungs. What it will do means it will maintain. It will not allow the more amount of sodium or water, and it will not allow the less amount. Like that it will function in the heart so that it can constantly maintain the blood pressure and blood flow. Obviously when the blood flow will increase, the blood pressure will be increased, right? So it will maintain or it will less the blood pressure or blood flow by maintaining the sodium and water intake from the other organs. Okay, so here the heart is nothing but atrial natriuretic tractor. And if we go on to the kidney, here you can see that aldosterones are nothing but the hormones that are present in the kidneys. So what is their role means actually what is the role of this aldosterone also? It is also relatable to this sodium and water. Why? Because it also maintains the sodium and water balance. Okay, sodium and water overflow into the kidneys. Okay, so what it will do, so to maintain the functioning of the kidneys properly, the aldosterone helps in the maintenance of sodium salt retention, means like sodium and water levels in the kidney. Okay, that is its function. So if we move on to the third one, gastrointestinal and adrenal cortex. Okay, so if you see that gastrointestinal tract, normally it contains the secretin, whereas the adrenal cortex erythropoietin. I'll explain you by showing the images here. If you see what is that, normally, so this gastrointestinal content, like the what is the secretin role is here. What is the role of secretin hormone? It will increase the physiological effects of every organelle in our body. Okay, in the hearts it will increase the heart contractibility, and in the kidneys it will help in the renal secretion, and in the liver it will also maintain the pancreatic secretion and everything, and it will allow the gastric juice, or it will also delay the gastric emptying, means it will always maintain the gastric juice inside by this secretin hormone, and it also helps in the regulation of thirst and satiety in the brain function. Okay, so like that the secretin hormone is having more roles in the like organelles itself. Okay, so if you move on to the erythropoietin that was given in the option here, erythropoietin is normally produced from the kidney itself. If you see here, what is the role of this erythropoietin means it is secreted from the kidney. What it will do, it will increase the red blood cell count in the bone marrows. Okay, so like that, this erythropoietin which is secreted from the kidneys, which will help in the secretions, like what they'll say, like it is not secretions, in the improvement or the increase of red blood cell count in the bone marrow. Okay, so that the bone can be strong. Okay, like that. If we move on to the next question, what is the next question is, this is also protein, it came from the enzymes itself, enzyme. So here you can see that enzyme, this is an enzyme. Now total enzyme, normally the enzymes will have two factors, like two parts, that is apoenzyme and coenzyme. Normally apoenzyme means nothing but it is a protein part of the enzyme. Okay, whereas the coenzyme is nothing but cofactor or inactive non-protein portion. Okay, so it is a cofactor that helps for the protein part. Okay, so when these two will combine, obviously they are individually inactive, but when they two will combine, they can form an enzyme. Okay, catalytically active holoenzyme. So here you can say that what is the protein part of enzyme is called as, it is known as apoenzyme. Okay, so like that, this apoenzyme and coenzyme will combine, when means without both any of them the enzyme cannot function, means it needs both the parts. So and then only the enzyme can function properly. So the apoenzyme is the protein part of the enzyme. Okay, we will move on to the last question from this video, that is nothing but which of the following is unit of productivity of ecosystem. Normally we know that the productivity of ecosystem is calculated like that. Net productivity means if we see the plant ecosystem, they'll absorb the solar energy and they'll show you some gross productivity through the photosynthesis process. In that they can also see that that in that gross productivity also they'll see that they'll show energy used for the cellular work and energy stored in the biomass. That is nothing but net primary productivity. Okay, so net primary productivity means which is produced in the biomass, and energy used is known as R here for the plants to survive. Okay, so normally what is the unit of these is, we know that we'll count this productivity or energy in the form of grams or kilograms per year. Okay, like that they'll count the net productivity of the ecosystem. Okay, that's it guys. So this section is over, and in the next video we are going to learn about some of the questions from this paper, and we'll end this video in another video itself. Means we'll complete this paper in the next session, so that after the next video we'll go on with the total paper revision, like I will add all these paper revision videos in a one playlist, so that you can watch the videos and analyze the questions that you have answered and everything. Why I am explaining like this, and already I told you before in the previous videos that you with only knowing the answer key you cannot understand the questions. If you know the explanation for that answer, obviously you can analyze, okay, why this answer is correct for this and like this. So that's why I am explaining the every question deeply, so that you can add to your playlist. And if you have any doubts, or if you are re-attempting this exam, it is helpful for you, and it is helpful for you in the next years also. Okay, like that. So if you want these questions, like the questions which I gave, if you want to know the answers for these questions, just try to watch my next video in the channel Biosastra Talks. And thank you guys. I hope you like the video. If you like it, just subscribe to my channel Biosastra Talks. And if you hope that this video is helpful for you, and then share it with your friends, and stay with me, and click the bell button so that you can get the notifications from the Biosastra Talks. Okay, thank you guys. Thank you so much.